Okay, welcome today, Michael. Thank you all for joining us for our live webinar as well, which will be presented live by Professor Michael Gelb, entitled Lipid Biomarkers for Newborn Screening of Treatable Metabolic Diseases, Discovery, Assay Development and Implementation. My name is Val O'Donnell. I'm based at Cardiff University in the UK. The webinar is being recorded and it'll be uploaded onto the YouTube channel, our LIPID MAPS YouTube channel, within the next 48 hours. We will be hearing Michael speak for about 45 minutes, and then that will allow 10 minutes at the end for questions. So please use the question and answer functionality as you think of questions. Don't wait till the end. Get them down so that I can then go through them in order, and then we will unmute you and you can ask questions yourself. Today's webinar is being sponsored by Merck. We're grateful for their ongoing support. And I'd now like to introduce our speaker. So Michael H. Gelb is Professor of Chemistry and Barbara L. Weinstein Endowed Chair in Chemistry, Adjunct Professor of Biochemistry at the University of Washington. He did his undergraduate training at UC Davis and his PhD with Stephen Sleegar in Yale University on the catalytic activity of cytochrome P450. Major work in the Gelb lab includes the discovery of protein prenylation, development of ICAT proteomic reagents, identification of phospholipases involved in lipid mediator generation, development of anti-parasite drugs, and development of mass spectrometry for newborn screening. He is the recipient of many awards, including the Repligen Award in Chemistry of Biological Processes from ACS, University of Washington Faculty Lecture Award, the Gustavus John Esselin Award from Harvard University, NIH Merit Award, and many others. I first met Michael last year in Nice at the French lipid conference Gelb, where he talked extensively about his exciting work in the area of newborn screening. For example, the use of newborn dried blood spots for detecting enzymes that metabolize gangliosides in neurodegenerative diseases. And I think that's what he's going to talk about today, judging by his title. And these mass spectrometry approaches for newborn screening of lysosomal storage diseases are changing how we detect and manage these diseases globally. His screening technology is being commercially developed by PerkinElmer, and an FDA-approved kit was developed and went on the market in 2016. He's had more than 40 new patents in the last 5 years on this newborn screening technology and has published more than 500 papers and 100 patents in biological chemistry. So we're really looking forward to hearing your talk today, Michael. So I'm going to hand over to you.
Okay. Oops. Thanks, Valerie. It's a pleasure to be here. Yeah, maybe most of you know I worked in the phospholipase A2 field for most of my career, and then something happened. My wife was pregnant with second child, and they did amniocentesis because of her age, and I asked kind of what they did, and they mentioned like Down syndrome and spina bifida, and I saw this movie Lorenzo's Oil about a metabolic disease and a father that tried to save his son's life, and since I'm an enzymologist, I began to think about testing for metabolic diseases, enzyme deficiencies, and I don't know, I sat on it for about 5 years, and then it occurred to me that mass spectrometry would be the way to go, that we can do many enzymes at the same time. And so I wanted to do hundreds of enzymes, and then I met Ron Scott, who is works at Seattle Children's Hospital, and he kind of gave me some reality that the goal is to screen not for every genetic disease, right, but for diseases that we have good treatments for. And let me go to the if we don't screen, the baby's born, you have rare disease, the baby becomes ill, they have no idea what's wrong. It might take months of diagnostic odyssey to figure out what's wrong, right? Because it's a rare disease, it's not your first choice on the list of possible diseases. And then by the time the baby is properly diagnosed, it's probably too late to treat the baby, right? So if only you had known at birth that the baby had the disease, you could have acted quickly and saved the baby's life. I think most of you know, like PKU, phenylketonuria, the lack of phenylalanine hydroxylase and the inability to break down phenylalanine, and this leads to a severe disease. But if you manipulate the diet and reduce phenylalanine intake early in life, you can prevent a lot of this. And that started newborn screening 50 years ago by Robert Guthrie, who was an MD in Buffalo, United States. You also have like galactosemia. You cannot give the baby mother's milk, too much lactose, too much galactose, missing a liver enzyme that isomerizes galactose to glucose, and so you reduce galactose intake early in life, and the babies do pretty well. But you need newborn screening, right? So there might be a sibling born with the disease, and then for the second sibling, you can be on the lookout. But if you want to save the first kid in the family, you need to screen pretty much everybody in the population. So these are rare diseases, so there's probably no known family history, in recent times. So basically, it's a public health program. You have to screen everybody. Yeah. Let's see if I can play this movie. I don't know if you can hear the sound, but you can read it.
The ability to walk, talk, swallow, eat, and see. Most kids with MLD won't survive beyond their 5th birthdays. In 2010, using stem cells derived from their patient's blood, researchers pioneered a treatment to repair the defective gene that causes MLD. Giovanni Price was the second child in the world to undergo this treatment. Today, Giovanni is 5 years old and has no symptoms of MLD. On August 24, 2015, Giovanni Price did something no child with this disease has ever done before. He walked to school for his first day of kindergarten.
Yeah, so that's a bit of this movie. You can see it on YouTube. It's called Miracle in Omaha. I'm going to tell you in a few minutes how this therapy works. It's of interest, certainly in England. It was just approved by the NHS in England. It's England's most expensive drug. But I'm not going to get into how much money should we spend on gene therapy. This question often comes up. I mean, this treatment probably cost £1.5 to £2 million. It's a one-time treatment. I wouldn't say it's curative, but you saw the video. These kids are going, to junior high school, and they've never lived more than 2 or 3 years without treatment, and they lose the ability to walk and talk and see and everything. These are horrible diseases, these leukodystrophies. I'm going to be talking a little bit about this disease, metachromatic leukodystrophy, or MLD. But basically, they're missing a lysosomal enzyme. You'll see it's a lipid modifying enzyme. So they take cord blood from the baby, which is baby's blood, right? It's placental blood on the baby's side, and they isolate with magnetic cell separation the hemopoietic stem cells, right? Some of these are floating around in the blood. They're not all in the bone marrow. So then the patient bone marrow is wiped out with chemotherapy. This is like bone marrow transplant that leukemia patients will get. The stem cells in vitro are modified with a lentivirus that carries the good gene for this enzyme and stably integrates it into the chromosome of these stem cells. The stem cells are infused back into baby's blood, where they repopulate the bone marrow, reconstitute the bone marrow, and then they start producing enzyme. Now, these are autologous, so the baby has no rejection of these cells. They're the baby's own cells. So that takes care of the peripheral body, but what about the brain? So it turns out some of these hemopoietic cells can cross the blood-brain barrier and reside in the brain as a component of what is called the microglial cells. And lysosomal enzymes have sort of unique ability that they're usually made in the ER of the Golgi on the ribosome, the ER, and then they're transferred to the Golgi, where they get modified in a way that targets them to lysosomes. the vesicles leave the Golgi and fuse with the lysosome, but sometimes the lysosomal enzyme leaves the Golgi and goes to the plasma membrane, gets secreted. So you'll find lysosomal enzymes outside of cells all of the time, but then they can be taken up by other cells by endocytosis, and then the enzyme ends up in the endosome, which then fuses with the lysosome. So it's been known for many years that lysosomal enzymes can travel from cell to cell through the extracellular fluid. So the microglial cells now armed with this enzyme, the enzyme can travel to cells in the brain that don't come from the bone marrow. And so you can get this sort of cross correction in the brain. So this is the 4th gene therapy ever approved in the world. The first one has been discontinued, so now we have 3. So this is LIPID, approved in Europe for MLD last year. There's a number of BBC documentaries on this treatment, cost, benefit, some of the science behind it. Anyways, I don't want to get into this politics, but the National Healthcare System Committee approved its use, and they thought this was, a good use of funding. Okay, so MLD, gene therapy, newborn screening. So when you test, so if you start this gene therapy on the affected kid that already has symptoms, it's too late to treat the kid. He does not benefit from the treatment. It has to be done pre-symptomatically. So if you want to save the first kid in the family, you need newborn screening, right? And that means you need to test every newborn in a public health newborn screening lab, because family history for these diseases is not adequate, except if the first kid dies, right? But you want to avoid that. So it obviously needs to be cheap and high throughput. So in the United States, for example, 50 to 60 diseases are included in newborn screening panels. In Europe, for reasons not clear to me, it's much fewer. England screens for about 12 conditions. Okay, so France even less, Italy more. And in England, you have now talk, for better or worse, you have Genomics England, where they're going to start sequencing 200,000 whole genomes of newborns looking for genetic diseases. This is a big topic that we think a lot about. I don't want to get into this today. Lots of challenges, lots of problems, lots of benefits. We're just going to see how it goes. It's going to be very rocky. There's a lot of people supporting it, a lot of people against it. I'm not going to go there today. So all of newborn screening in the world starts with dried blood spots. You may remember this if your parents taken from the baby, and we use a 3 mm punch from the spot, and this can contain biomarkers, enzymes, and we add buffer to rehydrate the blood spots, or we can extract the blood spots with solvent for lipid biomarkers, for example, that I'm going to focus on today. So I'm going to focus on the lipid side of newborn screening. We screen for lots of things that are not lipids, but actually a fair number of diseases are lipid based. So MLD, metachromatic leukodystrophy, is the deficiency of aryl sulfatase A, ARSA. All it does is removes the sulfate from this galactosyl sphingolipid. So you have a ceramide, you have a galactose sulfate to make galactosyl ceramide in the lysosome. The sulfatide is a major component of white matter, and so if you're missing this enzyme, you cannot recycle the myelin lipids, and the white matter dies. And it turns out most of your white matter is put on after you're born, so that's why we have a disease instead of affecting the fetus, right? That would not be a disease. That would be a terminal, a miscarriage. These are diseases, you can survive, the fetus can survive without this enzyme. So that's the only thing this enzyme does, is removes this sulfate to make galactosyl ceramide. So the obvious thing to do is you can measure the enzyme activity. We can do that, you'll see, or you just measure the accumulation of the substrate, the sulfatide itself. So this is on the left, you have 15 newborn blood spots from kids that went on to develop MLD. So some states in the US store the residual blood spots after birth, so we can go back and get find the patients that have the disease and get their newborn blood spots that have been stored and show that the sulfatide is elevated. But you see, it's not elevated enormously, and it starts to overlap with the reference range. This is random 2,000 newborns. So we set our cutoff below these 15 so as to not miss any of them, okay, to try to minimize the false negatives. But of course, we're going to have some false positives. So we need a second tier test. So the sulfatide actually is measured in 2 minutes by LC mass spec using a triple quad. You guys are all familiar with this. And we did a pilot study of 30,000 random newborns, and any newborn with sulfatide above this cutoff, we then reflexed to measure the enzyme directly. And we do that also by mass spec. We incubate the substrate with the blood spot extract and measure the product by mass spec using an internal standard. So we did 29,000 first tier sulfatides, and then 200 were above the cutoff, and then 198 of the 200 had ARSA activity greater than 30% of the normal. So this is not MLD. We think you need to have less than about 5 or 6% of normal enzyme before you start to have symptoms. You don't need 100% of the enzyme. We had one newborn at 8% and one at 0, and the DNA analysis showed that the 0% kid clearly has 2 well-known pathogenic mutations. This is an autosomal recessive disease. So we basically had no false positives out of 29,000, which is an essentially perfect method. But we need the second tier test. This has been published. So in the meantime, with the approval of the orchard therapy in Europe, and then we expect the FDA will approve this in the next year or two, there's been a worldwide increase in newborn screening for MLD, at least planning for it. So in Heather Brown at Manchester in England did a small pilot study. I don't know, England is very slow to add new conditions to their newborn screening panel. I don't know why. They're very conservative. So you have a family with MLD, they're pushing for newborn screening. They have foundations. You can read about this online. So Germany actually started to go live in 2 out of 12 regions of Germany. They now screen live for MLD. In France, a very large pilot study, Italy, etc. So New York. So this is moving fast. I want to talk about some other lipid metabolic diseases. This is Niemann-Pick disease. The enzyme that removes the phosphocholine from sphingomyelin to make this ceramide. You also have galactose on the ceramide. Removing that is Krabbe disease, and Gaucher is glucose. So we simply add we add surrogates of the natural substrate. They're almost the natural substrates, except the R groups here. Fatty acid groups are C8, C10, C12. So these are shorter than what you see in natural substrates. And so this, there would be ceramide otherwise in the blood spot. So we want to mark this enzymatic activity by using a shorter acyl chain. But these are essentially the natural substrates. Then we use internal standards that have deuterated R groups here. It's very simple. But we can do these all in the same analysis. It's mass spec. So it turns out, I want to talk about Krabbe disease. It's an interesting story. So the enzyme that removes galactose is a very serious leukodystrophy, very terrible disease. We can treat this pretty well now. So it turns out when you measure enzyme as your primary screen, if the enzyme is 0%, you're going to have the disease, but it's a continuum. So if you have more enzyme, you might have a later onset disease that affects you as a child or even as an adult. It's a continuum, and the activity becomes even higher than you have maybe no disease. But what does no disease mean? You might have slight disease. So it's a continuum. So when we measure the enzyme activity in dried blood spots, we would use a cutoff of, let's say, 10% of normal. And you get people near the 10% or even 6, 7, 8% that really don't have a disease. They have variants in their DNA that reduces the activity of the enzyme, but not to the point that it becomes pathogenic. And again, this is a continuum. So we always like to have a second tier biochemical test. And DNA is not a very good second tier biochemical test, as it turns out. You might think it's perfect if you just look for mutations, but we find often a lot of variants in the DNA that we don't know what it means. We've never seen these missense mutations before. It's a very complicated story, but DNA is not always reliable. So we actually look for this lipid called psychosine. Psychosine is galactosyl sphingosine, and it's very, very toxic to cells, and your body normally keeps this thing very low. The question is how is psychosine formed? This is a very interesting study. So this is galactosyl ceramide, and normally the GALC, the Krabbe enzyme, which is deficient in Krabbe, would take off the galactose and you make ceramide. And then acid ceramidase in the lysosome then follows and removes the fatty acid to make sphingosine plus fatty acid. But one hypothesis for psychosine generation is if GALC is deficient, acid ceramidase can act directly on galactosyl ceramide to make psychosine plus fatty acid. The problem with this is that acid ceramidase shows very low in vitro activity on galactosyl ceramide. It really prefers ceramide without the head group. But it turns out it does have a tiny amount of activity, and you can prove that acid ceramidase is generating the psychosine. The way you do this is it's really cool. If you knock out acid ceramidase in mice, you get a mimic of the human disease called Farber disease. And the symptoms are very different than if you knock out GALC, which is Krabbe disease. They're in the same breakdown pathway, but in different, one follows the other. So if you have a knockout of acid ceramidase, what's interesting, so if you have a knockout of GALC, you get psychosine, as I said. But if you knock out acid ceramidase on top of that, you don't get psychosine. So that proves that you need acid ceramidase to make psychosine. So when you knock out GALC, psychosine builds up. If you, on top of that, knock out acid ceramidase, you don't get psychosine. So you need acid ceramidase to make psychosine. What's interesting is the double knockout is less severe than when you knock out GALC alone. So when you knock out GALC alone, you have Krabbe disease, you build up psychosine. But when you knock out GALC and acid ceramidase, you don't make psychosine, and you get a disease that's much more mild and looks like Farber disease. So this proves that acid ceramidase is required to make psychosine, and it also proves pretty strongly that psychosine is the bad guy that causes the symptoms in Krabbe disease. The way we do newborn screening, so why don't we just measure psychosine first here for newborn screening if it's the best biomarker? Because it's a very difficult lipid to measure. You have to separate it from glucosyl sphingosine, which is called lyso-Gb1. The only difference is a hydroxyl orientation. So you need a long chromatography time. The amount of psychosine is low nanomolar, so you need a very sensitive machine. So there's just no way you're going to do this in a newborn screening lab on 1,000 samples a day. You'll never be able to keep your equipment clean enough to be at this sensitivity on a routine basis. So it's much, much easier to measure GALC. And then on 100 samples per year in New York where GALC is low, we measure psychosine in a second tier test. So that's the way we do it. And we have then essentially no false positives if we measure the enzyme and then psychosine. It's just not practical to measure psychosine first tier. The same thing for these other diseases. If you have Niemann-Pick disease, we measure the enzyme, but you also make lysosphingomyelin, which is sphingomyelin without the fatty acid. So we think acid ceramidase is responsible for generating lysosphingomyelin. We measure that as a second tier test. Same for Gaucher, we measure the enzyme. And then if you don't have the enzyme, acid ceramidase can act on the glucosyl, and we make glucosyl sphingosine, which is called lyso-Gb1, I think. And we measure that. This is an interesting story, side story. Lysosphingomyelin 509 was a novel biomarker discovered in Germany that's found in Niemann-Pick, and they didn't know the structure. The molecular weight was 509, so they call it lysosphingomyelin 509 in Niemann-Pick. So we figured out the structure. It's a novel class of, a new class of phospholipids. It's pretty exciting. It's N-fatty acylated serine O-phosphocholin. This is published 5 years ago. So we're making a bunch of these unusual phospholipids. I don't know their biosynthesis. We haven't really studied it. It's not really a priority. If you want to work on it, go for it. We don't know what the function of these lipids are. We know they're present in cells, and it's a new class of phospholipids. We have an FDA kit for all of this through Perk and Elmer that Valerie mentioned. Now I'll talk about another lipid disease. So you have a number of bile acid diseases, right? You don't need bile when you're a fetus because you're not digesting your own food. As soon as you're born, you need bile, right? The liver makes bile. There's a bunch of enzymes from cholesterol. And one of these diseases is called cerebrotendinous xanthomatosis, CTX. Very, very treatable by just giving bile as a dietary supplementation. It's essentially curative. But if you don't catch it early, these biosynthetic intermediates go into the brain and they're neurotoxic. It's a very serious disease. If you block this enzyme, it's a cytochrome P450 that puts a carboxyl group on the side chain of cholesterol, one of the steps in bile synthesis. What happens is the intermediate builds up. It gets an extra hydroxyl group on the cholesterol backbone by some sort of liver P450, we think, we're not sure. And then the side chain gets beta oxidized. Sorry, I misspoke. This is the bile pathway. The P450 puts a carboxyl group. Then in the peroxisome, it gets shortened. And this is quinodeoxycholic acid. You get another hydroxyl group shortened on the side chain, you get deoxycholic acid. But in CTX, you're missing this P450. And what happens is, so you keep the side chain, you don't put a carboxyl group here, you don't shorten it, you put a microsomal P450 OH here, and we see it as the glucuronide bioconjugate. This is the biomarker, the tetrol glucuronide, and it's elevated in CTX very nicely. So it's a very nice biomarker for CTX. It works perfectly. We did 30,000 newborns. We had only one with elevated marker, and it was confirmed to be CTX with no false positives. Lysosomal acid lipase, another lipid kind of thing. That's why I included today. This is the enzyme in your lysosome that hydrolyzes triglycerides, cholesterol esters and triglycerides that are part of your lipoproteins, right? That are taken up into lysosomes. And the severe deficiency of the lipase is called Wolman disease. And you can treat this by giving the missing enzyme. It's taken up into cells in the liver, into lysosomes. And we are not able to see the hydrolysis of the physiological substrates, the triglycerides and the cholesterol esters, in a 3 millimeter dried blood spot punch. So we had to make a more reactive substrate. These coumarin, these things are used in fluorescent assays a lot. It's a more reactive substrate. It has a phenoxy leaving group. You can measure this thing being hydrolyzed by the lipase, but it's not specific for lysosomal acid lipase. Other esterases in blood will hydrolyze this. That's a problem. So we made kind of a library of coumarin analogs, and we found that this methyl propyl is completely specific for the lipase, the lysosomal acid lipase. You can prove, the best way to prove that, the only way to prove that, or you can have a knockout mouse that doesn't have the lipase, but what you really want is a knockout human, and we have that. It's called Wolman disease. They don't have any enzyme, as far as we know, and they don't have any activity on this substrate. So we have the first specific substrate for lysosomal acid lipase. We can use this for newborn screening. Now, this is really where mass spec shines. The ability to do all of this in a multiplex assay. So we, inside of 2.1 minutes, we do an LC assay, 1 per newborn, where we measure all of these lipid biomarkers and other biomarkers, not just lipids, and we also measure enzyme products. So we either take the blood spot punch and incubate it with buffer with substrates, let the enzymes convert to product, and then use a chemically identical internal standard to measure the amount of product. That tells us the activity of the enzyme in a fixed time point assay, right? Or we can just measure these biomarkers that are present in the blood at time 0. We can extract the blood spot with methanol, chloroform, or methanol, and then combine this extract with the enzymatic product mixture, and then inject everything at the same time. Inside of 2.1 minutes, we can do we think 200 enzymes this way, but and 100 biomarkers. But we don't do all these because we don't have treatment for all these diseases. So we're not doing newborn screening for 200 diseases. The focus right now is on a set of about 20 diseases. Okay. And among those 20, about 8 of them have made it onto newborn screening panels in the United States, and a few in Holland and a few in Italy. Again, England and France are way behind for reasons that are not clear to me. So we have the ability to do lots of multiplex biochemical based newborn screening. So that's what I wanted to talk about today. The summary is, LC mass spec is a powerful technique for multiplex analysis of biomarkers and enzymatic activities for biochemical newborn screening of treatable genetic diseases. It's sufficiently practical and high throughput for population scale newborn screening. Newborn screening labs 30 years ago were using triple quad mass specs to measure all of the amino acids and fatty acylcarnitines in blood spots, to measure amino acidopathies like phenylketonuria, tyrosinemia. The fatty acid acylcarnitines, when they build up, are indications of mitochondrial beta oxidation diseases, the inability to metabolize fat. And these diseases are treated by minimizing fat intake into patients so that they never have to rely on beta oxidation to generate energy. And if they do that, then they build up these partially oxidized fatty acids, and that causes problems. So you can manage these patients with diet. Same thing for amino acidopathies. But now we're getting into more complicated, harder to treat diseases, lysosomal storage diseases, bile acid diseases. So now we're talking about diseases. In some cases, you can treat with diet, like the bile. Lysosomal diseases we're treating either with gene therapy or enzyme replacement therapy. You can give the missing lysosomal enzyme and it's taken up into cells. I think, in the next 10 years, I think there's 200 or 300, maybe more gene therapy clinical trials in the United States alone in the next currently. So we'll see what happens with gene therapy, right? It's a big maybe. So the MLD gene therapy looks remarkably good, approved in Europe. We probably can expect more of successful gene therapy in these, I would say, low-hanging fruit situations where you don't have to put the gene into every cell in the body. I think that's impossible right now, where you can repair the liver, or this trick of getting hematopoietic cells into the brain with bone marrow transplant. I think you'll see more and more of these gene therapies approved, and we're going to need newborn screening if they're going to be effective. I think sometimes the treatment without the screening is pretty useless, because you can't catch the disease in time unless you let the first sibling suffer and die. So I'd just like to end with my lab. I talked about all of these individuals' work today. I'm grateful to them. We collaborate obviously with lots of people all over the world for metabolic diseases, a lot of parent foundations. We're kind of the go-to lab in terms of if you have a disease that newborn screening is warranted, people come to me. We are the lab that has sort of delivered all of the new assays for newborn screening in the last 10 years. And we're working on additional diseases. I would say right now we're running out of diseases for which we have good treatment. So it's going to come down to gene therapy, I think, in the next 10 years. We'll see where that leads us. Okay, I think I'm done. Open it up for questions.
Thank you. That was really interesting, and the impact of that is enormous. It's just incredible. I mean, I'm really amazed to see a pilot study on 30,000 people using mass spectrometry. I've never heard anyone call that kind of number a pilot study before. I mean, that's a large cohort already, isn't it? And how long did it take to run those samples? I know it's a short assay, but even still, that's phenomenal, really.
So in my business, I get in trouble to call it a pilot study because, I mean, let me explain some semantics here. It's a little bit, it kind of, well, so officially a pilot study is when you have identified blood spots. You know who the patients are. And so to do that, you need parent consent, right? You can't screen, because if you have the samples identified, you can go to the family and say, hey, we found this disease, do you want treatment? And the parents need to know in advance that we're screening for that. Otherwise, you can't do it. So we call these pilot studies. In my lab, we're not allowed to do that. We don't have the parent consent. So 30,000 means de-identified. So when we find these diseases, we can't do anything about it, right? So there's some kid out there with MLD and we can't find him. We can't tell him. So now we have so why can't I do identified? Because I have no way to go around to all the birthing centers in Washington State and get parent consent. So now we have a problem. The federal government has an advisory panel that reports to the Secretary of Health and Human Services about what diseases should be added to the panel. And the states make their own decisions, but if the feds recommend a disease to be added to the panel, the states usually follow, at least many of the states, the progressive states, let's say. We have lots of states in the United States that are less progressive, right? Less funding for healthcare and things like that. So they say you cannot prove that your screening works until you find the patient and prove to me they have the disease. Now, that sounds like a pretty seductive argument, but it's wrong. In the case of, let's say, CTX, we have the biomarker is elevated, but it's only a biomarker, right? But it's the substrate for the enzyme that's elevated. But the DNA is super clear. So we know this kid has CTX. In fact, if we had data on an identified kid with this information, bile acid intermediate elevated and DNA signature of the disease clear, the clinicians would start treatment on the bile supplementation long before the symptoms occurred. So they would consider my data to be proof that we have the disease.
Yeah, that's really frustrating, isn't it?
Yeah. So we're kind of wondering, why do we need to find another one who's identified? And so we're fighting like hell with this federal panel to undo this rule that you have to find one kid in a identified pilot study, because we don't know how to do pilot studies in the United States. We can't seem to do them.
Yeah,
we can't seem to get the consent.
Okay,
but so back to your question.
Yeah, we have a couple of. Okay, go on
back
then.
To your question. I mean, we can do 500-600 samples a day on one machine at 2 minutes per baby. So we can do this in a matter of a few months, right? So
yeah, that's still pretty phenomenal. So we've got a question from Bill Griffiths, and then Alan Brash has got his hand up too. So Bill, you can unmute yourself and go ahead and speak now.
Okay, yeah, very, very interesting talk. So I just wonder if you see any value in looking at urine besides the blood spots. Maybe you want to look at urine perhaps for older children. So is that something you've explored?
Yeah, I mean, it's a very good question. I think urine provides, in some ways, a better sample than blood, because a lot of these built up metabolites don't accumulate in blood, right? They're passed into urine, right? And so typically urine would be used in diagnosis for some of these markers. And then in blood, we find that the levels are not very high sometimes. And the only reason we don't use urine is because newborn screening labs don't collect urine. And so it would, I'm told that it's a deal breaker that so we're dealing with state newborn screening labs, and so there's a lot of red tape and politics and resistance to change. And they are not interested in pursuing urine. The only place in the world I know that does urine for newborn screening is part of Quebec in Canada. So they have this history of doing urine. So yeah, urine would be a valuable addition. In the current climate, we're forced to use dried blood spots for no good reason other than nobody wants to change the rules and use urine. Yeah.
So actually, follow-up question with your dry blood spots. You just do I guess a solvent extraction and then a straight injection, or do you have a very short chromatography?
Yeah. So we try to keep the sample pre-mass spec steps to a minimum because this is high throughput and every liquid transfer matters. We typically just extract and shoot. So we could use flow injection in some cases, but in fact we do. But we're into diseases now where we need LC. We need LC for I would say two major reasons. Let's say aryl sulfatase A. So sometimes I didn't talk about I talked about aryl sulfatase A. Well, let me talk about the enzyme assay. So for aryl sulfatase A, we measure the ceramide, the galactosyl ceramide product after the sulfate is removed. If you do flow injection, there's a fair amount of the sulfate comes off in the heated ion source that's enzyme independent. And so we generate the product of the sulfatase in the ion source. So we need LC to separate the sulfatide from the desulfated product, because then we only integrate the desulfated product, right? But we get a signal in the sulfated substrate peak in the channel of the product because the sulfate comes off in the mass spec, but they separate. So that's one reason. And the other reason is the sulfatide concentration in blood is low enough that we really need the sensitivity gained by LC. We would never see it by flow injection. We need the sample to be concentrated into a tight window of time. So that's a case where we absolutely need LC before the mass spec. Sometimes we can use flow injection. But so labs right now are kind of using flow injection, but they're now expanding as we add on things like MLD. They're now just putting LCs onto their mass specs. That's a trivial thing to do because flow injection, you need a pump and an auto sampler. The LC just requires another pump and a column, right? Well, there's high pressure and leaking and clogging and all kinds of things, but this is not a problem. Illinois, for example, is doing 350,000 LC mass spec assays per year at 2 minutes each, and it works fine. Taiwan is doing this. So we have a situation where it works fine in a newborn screening lab. It needs to be done by LC mass spec, and the labs are coming around to realize that they're going to need to use LC for the future. So that's
one very, very final part. Is maybe you mentioned, is your assay multiplexed in the same way? So can you look at Wallman and CTX in the same assay?
Yes, everything I talked about today is done in 1 2-minute assay. And that's the beauty of mass spec. And we think we can do 200 enzymes and hundreds of biomarkers at the same time, but we don't have the need to do that right now. We don't have enough treatable diseases. So yeah, it's completely multiplexed. That's the beauty of mass spec, is it's completely multiplexed. Yeah. So I mean, we have some labs in the United States that have gone with fluorometric assays, let's say for certain lysosomal enzymes that are hydrolases, you can just look at a 4-MU, a coumarin come off a sugar and measure the fluorescence. That's fine. But now they're sort of all undoing their fluorescence and all switching to mass specs. Well, most of them started with mass spec in the first place because they saw the future of needing mass spec. But the ones with the fluorescence are now converting to mass spec because you cannot do things like CTX and MLD without mass spec, right? There's no fluorescent assay for these biomarkers. There's no fluorescent assay for sulfatide anytime soon, anything on the horizon. So I've been saying for 10 years, mass spec is the way to go. The fluorescence people came in for a little while, and I think now they're switching to mass spec. So pretty much most of the world doing this is now using mass spec.
Okay, thanks, Bill. Alan, you should be able to unmute yourself now.
Yeah, got it.
Perfect.
Yeah. Hey, Mike, that is really impressive, and you're doing a fantastic job for everybody there. I was amazed along the lines that Val was talking about, that you can handle 30,000 samples. I mean, this is a non-scientific question, but how do you actually manage that? There are thousands of things coming in the mail and have to be put into the machine and kept track of. How do you manage that?
Yeah, so I'm not a newborn screening lab for a living. We're a research lab, but yeah, they have things come in the mail by FedEx. They have a cutoff time at noon every day, and they like to be finished by 5 o'clock. They put the samples on the mass spec before they go home, and they have the data the next day. And so one of the things that is a challenge for LC mass spec is the software has to be really good. They cannot manually inspect all of the peak integrations by hand. So the software has to be really good at finding the peak. Or another important thing is you cannot have, I don't want to talk too much about integrating things that are not peaks, because Valerie knows a lot about that, but Alan does too. But we're talking about real peaks, big peaks, goes up, comes down, and no peaks nearby. Not too many peaks nearby. So the software always gets the right peak, right? So we have to be careful about that, just because we're integrating thousands of samples automatically. We can polarity switch. The bile acid we do in negative mode, the sulfatide in negative mode, but most of the other guys are in positive mode. But we can do that with if the negative guys come out in their own time window, we can be in negative mode on the fly and then switch back to positive mode. So the polarity switching works really well. As far as all the liquid handling, most labs have robots, but in our case, we were doing a few hundred samples a day. We just have multi-channel pipetters. It's no big deal. Now, they can make a mistake. So whenever they get a screen positive baby, they do go back and repunch the card and confirm they got the right baby, right? You can mix up babies. That doesn't happen for all of that stuff, but they know how to do all that, right? They're really good at it. So it's a pain, right? I mean, if somebody quits, you're in big trouble. You need to have backup people trained. They work on weekends. It's every day. They're doing hundreds of babies in every developed country in the world. They're doing this every day. So if somebody calls in sick, it's like a bank. Everything has to be backed up, right? You can understand that. They do it. That's what they do. Yeah.
So how many labs in the US have these capabilities and are actually running these types of analyses in practice?
So most labs have their own newborn screening lab. Some small states chain together. Let's see, Alan, are you in Tennessee still?
Yep.
So Tennessee has a newborn screening lab in Nashville, and they have they have 5 or 6 mass specs. Texas has 20 mass specs. California has 20 mass specs. So, Washington does Hawaii, and Iowa does Wyoming and stuff like that. New England, Massachusetts does 5 states in New England. PerkinElmer has a company that does Pennsylvania plus the military. So there are about 40 individual state newborn screening labs in the United States, each of them with 5 to a dozen triple quad mass spectrometers.
Yeah. Thank you very much.
Yeah,
Yes.
great. Thanks. So one or two scientific questions I've got to come now following on from that. So I noticed, I mean, did I notice this right, that the sphingolipidoses tend to be all neurological, like there's something specific about why losing enzymes in relation to sphingolipid metabolism always have massive neurological symptoms.
Yeah, I mean, Krabbe disease is neurological, but it also has a peripheral nerve disease, you know, these kids lose ability to walk.
Yeah, it's all neurological, isn't it?
So,
yeah.
No, but they have peripheral. Yeah. Oh, I see. Well,
Yeah.
well, in the case of, yeah, it's a good question. So, I mean, yeah, so I'm not a myelin expert. You know, myelin is mostly put on after you're born, and myelin has a lot of these sphingolipids and a lot of sulfatide. So yeah, no, so why, if you're missing lysosomal enzymes that would be universal in all cells, breaking down sphingolipids in all tissues.
I guess that's one part. Yeah, that's one aspect of it.
Yeah.
And the other aspect of it is that it seems like all of the ones causing serious neurological symptoms tend to be associated with sphingolipid metabolism. That there don't seem to be ones targeting phospholipid metabolism or fatty acid metabolism, are there, that are neurological? It seems to be
Yeah,
more often than not a
that's a good
sphingolipid
point.
related.
Yeah.
Yeah.
So I don't know. My stupid answer is, you know, myelin has a lot of sphingolipids.
Yeah, but and then how do we think
So
about gene therapy?
Tay-Sachs, you know, it's a ganglioside disease. I don't think that's a white matter. It's not a leukodystrophy, right? So for some reason, I guess myelin, you have a lot of breakdown of sphingolipids that have relatively short carbohydrate head groups. You know, not gangliosides. Gangliosides, you know, more Tay-Sachs is more neurological, less white matter. And, you know, you have GM1 gangliosidosis, which you're removing a galactose from gangliosides, right? Or Tay-Sachs is the hexosaminidase. These are, you know, far away from the lipid. So these white matter diseases tend to be enzymes that work near the lipid, right? Near the ceramide. Yeah.
But it's quite striking, isn't it?
Yeah, but then you have Fabry disease, which Fabry disease takes the galactose off of galactose, glucose, ceramide, and it's not very neurological. And so you have an enzyme that takes galactose off, it's neurological. You have an enzyme that takes, galactose. Yeah, you have 2 sugars on the ceramide. Fabry, you have 3 sugars on the ceramide. You take off the non-reducing end first sugar of 3, you have Fabry disease, which is not neurological. You take off the sugar on the ceramide, that's Gaucher and Krabbe. These are neurological. So it depends how close you are to the lipid, and I don't understand that.
Yeah, it's interesting. I mean, that's a real challenge then for gene therapy, really, isn't it?
Yeah. So gene therapy for neurological diseases, you know, there are 2 approaches. One is the stem cell approach, where you put in the gene permanently with a lentivirus, and then these hemopoietic cells, some of them go into the brain, and cross correction is critical. So this only works for lysosomal enzymes, where the enzyme can be released from the cell and then taken up by other cells that don't come from the bone marrow.
Okay.
You know, so lysosomal enzymes, that's critical. But you're not gonna, an enzyme that's in the cytosol in the brain, you're not going to treat this with gene therapy. But they start injecting viruses into brain ventricles, and maybe they can put enzyme into the cytosol of brain cells. But this is much harder to do. So the low-hanging fruit in gene therapy would be things like lysosomal storage diseases. Also, you know, hemophilia, where you transplant liver cells, right? Immunodeficiencies, where you can't make T cells. You take out the baby's stem cells, put in the gene, and put it back in. Now you make T cells. So you can transplant the blood, right? You can't transplant the brain so easily, right? So
I guess since Alan is still there, how about skin? For, you know, Alan knows a lot about genetic disorders of lipid metabolism in the skin. So are they being targeted with gene therapy, Alan? You can still unmute yourself if you're there.
Yeah. I think not, although in principle you could say it could be done. Obviously, you could even apply it topically, but then you've got a large area to cover. So I think that's not being done at the moment.
Okay, so we got one last question before we finish up. So Matthew Henderson, I need to give you permission to, okay, you can unmute yourself now, Matthew. You've got a comment or a question there?
I'm not sure if you can hear me.
Yeah, we can. Go for it.
Oh, it was more of a comment in response to your question about the direct link between mucopolysaccharidoses and neurological symptoms. And I just made a comment that neurological symptoms are quite common in many different inborn errors, sometimes vasculopathies, fatty acid oxidation defects. That's a common sort of scenario. So more of a comment than a question. Michael's talk was excellent now that I've got a chance to speak. Thanks.
Thank you for that. Okay. Well,
I tried to, for example, we work a lot on the mucopolysaccharidoses. These are the enzymes, the lysosomal enzymes that break down the glycosaminoglycans that are present on, cell surfaces, and these are well treatable by enzyme replacement therapy. But I didn't focus on that. This becomes now carbohydrate analysis. There's no lipid. We're looking at the enzyme first, and then we measure the accumulation of the glycosaminoglycan biomarkers. And this is a whole nother story. But we have really good biomarkers for glycosaminoglycans. We don't analyze the whole polymer; they're too complicated. We look for small fragments that are produced endogenously. Always by mass spec. Mass spec is so powerful, as you know, for metabolite analysis in general.
Yeah.
So.
Okay. Well, I think we're just coming up to 6 now, so the timing is perfect to end on that note. And thank you very much, Michael, for an excellent talk and absolutely heroic and fantastic work that you're doing for these families and children with these terrible diseases. Thanks, everyone. See you for the next webinar.
Thanks, Valerie.